Now we have a presentation from the Minnesota Mycological Society's scientific advisor, Dr. Peter Kennedy. He's a professor at the University of Minnesota, and he is the director of the Kennedy Lab in the College of Biological Sciences. So Peter, if you're ready to take it away, I really look forward to your presentation.
Sure, yeah. Well, thank you guys all for this opportunity to talk to you virtually. It's something I get to do usually about once a year. I was gone last year on sabbatical, and I'm glad to be back. And I thought it'd be fun to talk about some fungi that I sort of ran across almost accidentally. And I'll talk to you guys about ways that I think you can help us as club members hopefully make the records of these fungi better, I guess, documented and more permanent by thinking about including them in the collections we have at the Bell Museum. So I'm going to go ahead and share screen. I don't know, honestly, if I'm going to get through all 21 of these fungi. I picked the number 21 mainly because we're in the year 2021. But I'm very happy to share this set of slides with the club and to have it posted on the web. Nan, you can check and make sure, but I'm pretty sure that I've got all the proper crediting of images from various places. So I hope we're not in trouble if this gets shared publicly. Otherwise, I will take the full blame. So here we go. So I'm actually gonna, before I jump into kind of who these 21 fungi are, I wanna give you a little background on how I came up with this list. And so I'm gonna start a little big. Sorry, it's a little bit sort of, I guess maybe not surprising given that, you know, this is what I do in terms of teaching and being a professor, that I'm going to start here, but I'll do that, and I promise we'll get to the fungi and the cool pictures of them very soon. So I think a lot of you guys have heard this term before, this idea of biodiversity. It's this big term that we as scientists like to use. It's a broad term. This is something that, you know, can be defined in many ways. I actually had some fun when I looked this term up on the web. So the definition for this is the study of the variability and variety of life on Earth. And if you go to the Wikipedia page, there's actually a picture of mushrooms right on the Wikipedia page for biodiversity. And so I thought that was a pretty neat connection given our common interest in these fungi. So when we think about this term, think about biodiversity, sort of what we're really interested in as scientists, or, you know, sort of anybody else, is thinking about all the different types of organisms that we have on this planet. And for this club, right, we're thinking a lot about the fungi. So if we think about the way that people have thought about biodiversity and studied biodiversity for a long time, the main way that it's been done, at least from a scientific perspective, is to collect and store representative samples of different species in museum collections. So this is a picture of one, but there are many different museum collections that exist across the world. We in Minnesota have a museum, the Bell Museum of Natural History. If you haven't gotten a chance to see the beautiful new building that is over next to the Twin Cities St. Paul campus, unfortunately with COVID, things have been slowed down dramatically in terms of visitation. That said, it opened just a few years ago and it's got a huge number of visitors. So it's been very, very popular since it was open to the public back in 2018. That said, it's actually a place where you can go and you can learn a lot about the science and the natural history of our state, but it doesn't have any of the collections that you might think of when you think about a museum in the context of what I just mentioned. So in actually one of the buildings that's on the St. Paul campus, we have a large number of collections, and the collections where the fungi are held are in the herbarium. Well, some might say the fungarium. But the Bell Museum herbarium includes the algal collections, the mosses that Tim was saying, and rightfully so, quite beautifully in many pictures today, are there. In the bryophytes, we've got lichens and fungi. So both of those would fall under the purview of folks that are interested in fungi. There's some paleobotany collections, and there's lots and lots of plants. There's almost 1 million specimens in that collection alone. And so it's really a kind of an amazing place to think about biodiversity. And these collections, while many of them local to Minnesota, come from all over the world. So if we could, go into the building to see where these collections are housed, you would see something that looks like this. And if we think about the fungal collections, there's about 225,000 fungal collections that we have in the Bell Museum. Of the fungi that I think a lot of you guys are excited about, and I'm most excited about, the macrofungi, there's about 72,000 collections that are made. And then there are many, many lichens as well, almost double as many. And those are held in various locations within the collections. So I just want to say that there is a way that you can still learn about these collections from the comfort of your couch, even if we can't go in and see these. You can check this all out. There's this really, I would say, fantastic website that's been built over the years by the curators and the team at the Bell. It's called Mapping Biodiversity. It's the Bell Biodiversity Atlas, and you can go in and you can click in there and search collections. You can click on the fungal collections and you can type in your favorite fungus and you can see, do we have a record of it? And so this is a way where we've kind of tried to open the doors of this collection into sort of a much more public forum and let people know about what kinds of collections are there. So that's the collections, and that's sort of the side that I've been involved in to some extent in my role at the University of Minnesota. I do want to acknowledge MMS. MMS has been a very kind partner to the Bell and to the Bell collections. There's been a generous donation from the MMS to help with supporting some of the curatorial activity in the past few years, and we're certainly grateful for that. MMS has also donated some funds to help with sequencing some of the specimens that collected through the MN Fungi project, and hopefully those specimens are now going to make their way slowly but surely into the Bell collections now that I'm back from sabbatical and ready to try and help the MN Fungi team make some of those collections have their official home in the Bell collections. So that's sort of the science side of thinking about this topic, but there are lots and lots of people, including many people here at this presentation, and certainly lots of other folks that are interested in collecting biodiversity data as well. And so some fun facts about that. So you can look in various places, but sort of a shockingly big number. In the United States, it looks like many, many people are out there in the wild, looking at all kinds of different aspects of nature. So in this case, maybe it's just wildlife, but I'm sure that this can count for fungi as well. And what's been exciting is that there's been this real revolution in the ability to share those observations through various online portals. And so one of the biggest ones that's really taken off and done well over the last, say, decade or so is something called iNaturalist. And so you can click on this link, or you could just type that into your search bar, and what you'll find basically this pretty amazing data-rich website. You can log in, it's free. You can install this as an app on your phone, but you can see here that there's almost 60 million observations that exist of records of people taking some picture of some organism on this planet and documenting where they were when they took it, and then often through sort of community effort trying to put some name on what that organism is. And so conveniently, they've made this so that you can sort and search and actually download the data from iNaturalist. And again, it's freely available. And so if you do that, I thought it would be fun to say, well, what if we do that for fungi in Minnesota? And if you do that for fungi in Minnesota, I think we're actually doing pretty well. The fact this is, again, only been about a decade or so's worth of observations, but there's about 10,000 records of fungal collections or specimens that exist in Minnesota, almost 1,000 different identifiers, almost 2,000 people making those observations, over 740 species. So a good range of information that exists here. So the question that came up for me, again, sort of, like I said, in a way where I was surprised because I had seen something on iNaturalist and was quite sure that it was in the Bell Museum collections. And when I went and searched the Bell Museum collections, I couldn't find it. And that surprised me. And so it led me to this question of basically, we've got these two resources, both interested in studying and documenting the biodiversity and the fungal diversity of our state in Minnesota. Is there things we can learn from one that could help us actually inform the other? And so in this case, we're going to think about some of the collections or the observations that have been made in iNaturalist that don't have representation in the Bell Museum collections. So this gets us to the 21 most wanted fungi in Minnesota in 2021. So just to give you, I won't bore you with the details of actually trying to get basically the two lists of species to line up and doing all the other things that, sort of sorted these, dealing with some nomenclature issues, etc. What I was going for was a list of fungal species that had been observed or identified as present in Minnesota on iNaturalist that I couldn't find when I searched the fungal collections for the Bell. I thought it would be good to kind of have a range. So this is a select set. This is not, there's not just 21 of these. There are actually hundreds of mushroom types. I'll explain more about that in a second. Collectible in different seasons, thinking that you guys hopefully are going to be out in the woods sooner than later in the spring, summer, and fall looking for fungi. And then also as an opportunity to talk a little bit about some of the poisonous and edible mushrooms that exist in our state. So my goal, I guess my ask with this presentation is that if you happen to encounter any of these 21 species, it would be great if you would be willing to do what you saw in those beautiful pictures that were taken by many of the club members and won awards. Take lots of pictures in the field, know where you made those collections, take a good set of notes on, the habitat, where it was found, what was growing on, etc. And then if you're willing to share that and donate that to the Bell Museum so that we can get this permanently included in the record of the Bell, it will be something that can be basically held in perpetuity, studied scientifically, and accessible to sort of all those who might be interested in knowing more about our friends, the fungi. So here's the kind of schematic. We're going to bounce around on the schematic of the different groups of fungi. So sort of working off of the common names. I know many of you are coming from all kinds of different backgrounds. I see some folks that are probably know many more fungi than I do that are on this call, to folks that may be at their first MMS meeting and may be interested in only thinking about fungi that, they know from common names. And so I'll do my best to kind of try to meet you guys where you're at, but I'll try and keep you oriented by using this as our guide. So we're going to start off with a set of fungi that hopefully, if I can get this to cooperate, are the ones that we might sort of classically think of as the mushroom group. And so these are the guild fungi. And so the first fungus that stood out as one that was, again, in iNaturalist, and also let me clarify, I did not anonymize who the collectors were because this is public data. That said, this is not my effort to try and say, the identification is in any way right or wrong on iNaturalist, with one caveat at the very end where I think there may be some reasons to think that the identification wasn't right. But I was consistently impressed with how well the iNaturalist community was able to put what I think are accurate scientific names on a lot of different fungal specimens. And so kudos to all the people who are going out into the woods, taking pictures and posting to these public locations. I really am impressed with the community effort that exists and excited to try and use this to help us document more of these fungi from again, sort of a scientific specimen-based perspective as well. So this is an Amanita species. It's the carrot-footed Amanita, or also known as the carrot-footed Lepidella. It's Amanita docipes, which actually the Latin name is the carrot-footed Amanita. Careful with Amanitas. I would say that I could not find a record for whether or not this was an edible species or not. That said, I would say please avoid Amanitas as a general rule from any sort of consumption. Many of them are, in fact, quite poisonous, and there are a number of the quite poisonous ones that are common in our state. That said, this is a pretty distinctive Amanita. So it's a big one. It's whitish. It's got a really warty top to the cap. So those are remnants of the veil, the little sort of mushroom that starts off in the ground wrapped in this one layer of tissue. And then sort of as it spreads out, those remnants stay on the cap. You can see that. Interestingly, two other big features. So importantly with mushrooms, you should, when you're learning to, collect these, if you haven't known this already, make sure that when you look at this, you collect from the base, and you may need to go down into the soil to remove some of what's underneath it to make sure you've got all of the specimen. In this case, you got this huge bulb, this interesting sort of orange staining bulb that is pretty distinctive within Amanitas. So that's a character that's important. And the smell of this sounds interesting. I have never encountered this mushroom, but according to Michael Kuo, who runs the website Mushroom Expert, it sounds like it's a very interesting combination. It says here, sweetness and foulness with a hint of rotting meat and chlorine. So another thing to pay attention to in these slides, I've tried to make these sort of, hopefully, sort of a range of characters to pay attention to. Where you find your mushroom matters. So these ones grow on soil. What type of habitat you're in. A lot of these are going to grow in forests, but in different forest types associated with different types of trees. So that's important. And then to some extent, the phenology matters. So these are mushrooms that you're probably going to find not as the first mushrooms in the early spring, but as mushrooms that you would see later in the season, in the later summer or the fall. So that's mushroom number 1. More information on that one exists on the Mushroom Expert website. Second mushroom is Entoloma vernum. Vernum, the Latin name for that is spring-like. And so this is a spring mushroom. This is an interesting species. You can actually see here, the picture is a painting. So it turns out that the study of this, Michael Coe's done a bunch of work on this. Probably this is a species complex, but in field guides and other places, you will find this. These are relatively small mushrooms. They're neat because they have pinkish colored gills. They also have a pinkish colored spore prints. They have this button-like top. So they're sort of a gray-brown, and they have this little sort of umbonate top. So that's distinctive. They have a little bit of white mycelium at the base of the stalk. And hopefully, if any of you are interested or have, the capacity to, the spores of these things are just fascinating. So when you think about mushroom spores, usually we think about these, sort of nice balloon-like structures. These ones have many, many different sides, 6 or 7 different sides. And that's a characteristic that we often see in this genus. So this is also known by the name Nolania verna, but it looks like Entoloma vernum is the right name. So you'll find this in the spring. If you're looking for morels, keep an eye out for this one. Both hardwood and conifer forests, and you'll find it on soil. Okay, so the last of the guild mushrooms, interestingly, this is a different one. So this is Panis neostrigosus. A little hard to see from this picture. A little bit better if you look at these pictures. So Panis is one that has some relatives that you may have seen. There's some actually that glow in the dark. Not known to be edible, so this is unpalatable. It's got a kind of purple-brown color to it with some pubescence, a little hair on the upper cap surface. And then it's got gills on the undersurface. But interestingly, if you look at this and you look at this relationship of this particular group of mushrooms to all the other mushrooms that are out there, turns out that this is in a group that belongs to the polypores, but has basically, in this case, it has gills rather than tubes. So sort of an interesting fact about that. You're going to find this on logs. So there's going to be a lot of wood-associated fungi. I figured these would be ones where when you're walking through the woods, you might happen to run into a downed log or look at a stump or otherwise. Fruits on dead hardwoods, for what I can tell. So probably not in the conifer forests. And it sounds like it's kind of one you might see almost all season. So keep an eye out for it. Okay, so we're going to move over to the boletes now. And so this is where, boy, I've already forgotten those beautiful toads' name, Judy and somebody. We're sitting on top of a beautiful bolete in John and Claudette's picture. That was really nice. So this is a species that is interesting. It's called Chalciporus rubinellus. It's a beautiful red-pored bolete. Red-pored boletes are not particularly common. You tend to see a lot that have yellow pores. You see some that have cream-colored pores. But this one is really red and really distinctive. You can see it in both of the pictures that were taken. So I'm going to tell you a little bit about this one. We don't know about the edibility of this one. So I would not suggest when you collect this that you save a little sort of set that you might want to try to eat, because red-pored boletes are known to be toxic. There's a handful of them that you really need to stay away from. So I would suggest not one to consume. Relatively smaller size, beautiful. I mean, just amazing coloration. The red and the yellow are really distinctive. The red pores are quite key. Has a very yellow stalk if you break open the stalk. Unlike a close relative, there's another Chalciporus species that we have in Minnesota. This one does not taste peppery at all. So if you take a little teeny bit of the cap, just a really small amount, you chew on it for 10 or 15 seconds and you spit it out, that's going to tell you enough to whether or not you're going to taste anything that tastes peppery on your tongue or whether it's not. And that will help you distinguish those two. Turns out there's a close relative to this one. This is Chalciporus pseudo-rubinellus that has more of a yellow mycelium that's on the stalk. This may be a more southern species than we're used to seeing, but that picture was, if you look back at the slide there, this is pretty far up on the, I think on the North Shore here. And so I think we've got this one in Minnesota and actually multiple locations. I'd love to see this added to the collections. Okay, moving along. So that was one of the boletes. So let's talk about the teeth fungi. The teeth fungi have some of the best common names of all the fungi that we're going to talk about tonight. So we'll start off with this guy. This was actually the one that kind of made this whole presentation come to life. I was looking for a record of Hydnellum peckii, and I could not find one in the Bell collections. That really surprised me, because I think this is one that it's actually been seen multiple times on iNaturalist. It's quite distinctive. It really doesn't look like anything else when you encounter this thing. It's got these big, Tim said, guttation, this sort of interesting droplet form, these bright red droplets on this otherwise white mycelium. What's cool about this is that's the top of these. The bottom are actually covered in these teeth-like structures. That's where the spores are going to be released off of this mushroom. Supposedly this is edible, but unpalatable, according to the records that I read. So I don't think this is, again, another one for the pot. It's got a pale upper surface matched with these beautiful red droplets, hence some of the nicknames. One of the most common ones that's out there is strawberries and cream. It's a very nice blog on this particular fungus in the Forest Flora Narrative. If you look at that blog that I've posted at the bottom, you can read a little bit about this. This is where some of these pictures come from. It will turn a little bit more dingy brown with age, and so it's maybe not going to be as distinctive as it is in this young form. Again, it's got these nice sort of light colored teeth that are on the undersurface, important characteristic, typically in conifer forests. And so that's one to keep track of, probably a species that exists also in Europe, not just in North America. And you see this one in the summers and in the falls. So keep an eye out for this one as the mushroom season of 2021 gets longer. So here's another tooth fungus. This is the spruce tooth, Phelidon violescens, or violescens. It is another one that, again, you might, when you first see it on top, sort of, you flip it over, you see the teeth. That's distinctive. They've got these light colored teeth. There's some nice pictures. This is one that looks like it's definitely present north of us. Some folks in Canada, so the Myco Quebec Club has a fantastic set of myco images that are up, and this is where I found these images. So it's got this pattern, a little bit of zonation on the upper surface of the cap. You can see that in the bottom picture there. It's going to bruise a little bit. It's going to turn a little brown with handling. Supposedly a fragrance, pleasant odor to it. Interestingly, spiny spores if you get it under a microscope. And it says the name for this, violescens, where it comes from, the brownness colorations can be grayish brown with a hint of smoky violet, hence the species name. So sort of a fun way to understand why it got that Latin name. It also goes in certain field guides under the name Bankera violescens. So this one looks like it's really a spruce associate. So this is where you're going to be looking in spruce forest, probably, up in the northern part of the state. Maybe Gene and the Paul Bunyan Club can help us out here. But this is one that would be great to add into the Bell collections. Okay, so we're on to the false morels. So I'm not going to, unfortunately, for those of you who came to this talk hoping to see true morels, I'm not going to talk about true morels. I know that the club will have some true morel talks coming soon, but let's talk about the false morels because they're important. So these are sort of morel lookalikes. This one is Gyromitra brunea. It is a species, the coloration on this photo maybe is a little different than the coloration that is suggested to be what is a distinguishing feature of this Gyromitra species. So this one really has more of sort of an amber, reddish color. It has sort of a structure that, unlike some Gyromitras that look like just a giant brain, this one actually is a little more Helvella-like, where it sort of folds into something saddle-like. It's got a pale set of undersurface, and this, again, sort of tawny reddish brown color. The spores of these look pretty amazing. So if you could get this under the scope, if you cut this in half, it's basically quite porous, so it doesn't have a thick, solid stem. And it's got these interesting little points at the end of the spore. So it's got the sheath and these little apices that are there, large droplet in the middle, large oil droplet there. It's known sometimes in field guides as Gyromitra fastigiata. You might see it under that name. That is a European species and a European name that should stay in Europe. And so I think Gyromitra brunea is what we should call our North American version of this. This is a spring mushroom. So when you're out looking for morels, be careful. There are false morels out much of the same time, but it'd be great to get some of these false morels into the bell. Another in that group, so this is Gyromitra perlata, a little bit different in the morphology. In this case, the stature of it is changed, but the common name for this one, pig's ears, I think pretty appropriate given what it looks like. Here are some other pictures of it. According to Michael Kuo, so again, poisonous, stay away from Gyromitras. Gyromitra perlata can be separated from similar cup-shaped species by a combination of features, so a reddish to dark brown, spores that have these pointed apiculae, which are the projections that stick out, and then the coloration, again, tends to be different. So you either have an orange brown or a yellow brown, or you have something different in terms of the spores. I'm not expecting you guys to, if you think you have this mushroom, necessarily look at these microscopically. If you're willing to donate the collections, I'll happily do the microscopy work to see if we can verify your identification. Looks like a couple others. So there's another species that we have in this area that in this case has more of a pseudostem, more red, different features. This is Discoides venosa. You'll find this near wood. So this is rotting wood, dead wood. That's kind of the habitat you'll see. But again, fruiting in the spring. So keep an eye out for these Gyromitra species. All right, so we're going for now the shelf fungi. So these are the polypores. So I'm gonna keep going. So this is one that I think, honestly, people have seen, and I was really, again, surprised we did not have this in the collections. So this is Marypilus sumstensinae. This is something called the black staining polypore. For those of you that are fans of hen of the woods, this one is kind of similar habitat, similar look to it, especially when it, in this case, is young, before it ages and gets that really sort of characteristically black staining that's different than what you would see on most hen of the woods. Inedible, according to Tim Baroni's guide, when young and tender, but hen of the woods is better tasting, according to Tim. Large, so these are big. This is a sizable, mushroom that you're going to find. They start off sort of whitish, but they'll stain black. So that's going to be characteristic. You have very small tubes that are on the underside of the surface. So no gills, no teeth. In this case, we have one collection back from the early 2000s of something called Marypilus giganteus in the Bell Herbarium. It looks like that's a name that has been misapplied. And so it would be great for us to add in this collection. That seems to be a European species. This seems to be the North American one. You'll find this fruiting at the base of dead trees. So keep an eye out for dead hardwood trees fruiting in the fall, and this might be one that we can add to the collection. This is an interesting one. So this is called the blushing rosette. You'll see why in just a second. A little hard to see in this picture exactly what we've got here, but these pictures on Michael Kuo's page I think are great. So similar habitat. You're looking at the bases of trees. Probably not an edible, or at least not one of desirable texture. Sort of brownish on the upper surface, a little bit of sort of fine hair, whitish on the inner surface. You'll see pores, may have a little bit of a pseudostem. Bruises a little bit, will turn a little bit red when handled. Sometimes it can have a little bit of a what they say is a pink or red juice that comes out. Interestingly, it comes in two forms. So just like our friend who got featured in the pictures there for Entoloma abortivum, right? There is a second morph of this, basically same species, and the other morph, the aborted morph, in this case, is this just strange multi-headed growth that has lobes that are sort of, at this point, brown and sort of tomentose, and so there's some hair on top of these. It would be awesome to get collections of both representative morphs in the Bell collection itself. So you'll find these, again, near dead trees, could emerge from some buried roots, so you might find it out, away from where a tree has died. Tends to be a summer or fall fruiter. Okay, so a little more, smaller, but again, distinctive. I was trying my best to give you mushrooms that I feel like you'd have a fighting chance of saying, wow, that looks distinctive enough that I think I could sort of look through the guides and sort of see what this looks like. So Climacodon pulcherissimus is a species that is interesting because it's very hairy on the top and it's got an interesting looking undersurface. And so it almost looks like it's sort of hairy on both sides. So it's white on top, cream colored, matted, woolly, hairy. So going to be distinctive in that way. And it's got these fine teeth-like structures. And they are going to change a little bit in terms of coloration with age. They'll sort of mat up and come together like, you know, a dog that hasn't gotten a bath in a few weeks. If you happen to have access to a little bit of, in this case, potassium hydroxide, this might be something that in the foray, when the ID tables come back, you get a dropper bottle out, you get a red reaction. So it's a distinctive character that helps you, again, identify who this is. It tends to be growing on mostly hardwoods again. So we'll find it in, you know, the oak forest or the mixed hardwood forest that we have a lot of around the Twin Cities or throughout Minnesota. You can find these in shelves or in clusters. You can also find them growing alone. You'll see this one hopefully sometime this summer. I've made a collection of this. I haven't gotten into the bell yet. So I'd rather have you guys bring in a collection, but I think we can get this one into the bell sooner than later. This is a new fungus to me, the marshmallow polypore. This is a picture of the collection that was taken, and you can see what it looks like on here. I'm going to zoom over to the picture where I can see the name just a little bit better here. So Spongiopellis pachyodon, and it is, again, one of these interesting polypores. So on wood, whitish upper surface, teeth-like structures, more flat, plate-like, slightly different than the sort of individual teeth that we saw on that Climacodon species last time. Not going to stain, so we're not going to have any of those chemical reactions. Different than a couple of relatives, so a different Climacodon species. You might see something that you think is Urpex lacteus is the species that looks like this. I was talking to a buddy of mine, Jonathan Schilling, who's here, a professor at the University of Minnesota, and he was saying you'd find Urpex a lot on branches and twigs, pretty common. This one you're going to find more on the stems of trees. It's actually causing a rot of the heartwood in the tree, and so you'll find this on trees that are alive. So it's a pathogen, often on oak, and it looks like you find this one potentially in these sort of shelf-like clusters, which will be a feature that would be interesting and distinctive. So you getting this off the tree, you might need a hatchet or a little more than a mushroom knife to sort of pry this if you get a reasonably sized collection. Fruits in the summer and the fall. This is I had a little trouble sort of fully believing this picture, but the false turkey tail that was actually featured in the mushroom pictures, one of the first pictures that we saw. It's in this group, Sterium. So this is Sterium ocratioflavum. What's neat about this is if you look at the picture, so a colleague and friend of mine who is actually a friend of MMS, so Patrick Leacock, who gave a talk in the fall. Patrick's website, the MycoGuide website, has an identification guide to different Sterium species in the Chicago region. And this one he has listed as well. You can see these really erect hairs. So although this is a polypore, it's very hard to see the pores. They're really, really, really teeny. So that brown surface is actually where the pores are. The upper surface has these funny looking sort of erect, whitish looking hairs. There's other Steriums that exist. There's other that have hairs. So here's a picture of Sterium hirsutum. But this one looks distinctive enough that I think we could feel pretty confident that this is ocratioflavium rather than some of the other Steriums that we might find here in Minnesota. Looks like it grows on hardwoods. Sounds like it's rare in the Chicago region. I have no idea about its rareness in Minnesota. I liked the name of this one. I just thought it was great. Giraffe spots. So this is Peniophora albobatida. And Peniophora albobatida it's a crust fungus. So again, in this sort of kind of polypore group where you can actually got these little pores where the spores are coming out of, but it's going to grow right directly on the surface of whatever the logs are that it's growing on. So it's got a crust-like body and it's got this brown and white combination that's pretty distinctive. A lot of the crusts are really hard to identify without spending a lot of time under the microscope or grinding these up and getting their DNA. But this one looks like one that you might be able to pick out with basically the characters that we have here. The name, the Latin name again, is kind of neat here. We've named this basically because of the white and the reddish brown patterning. And so the albo and the badia is the sort of giraffe spots. And you can see here, actually, that's a thorn that's coming out of the tree, and the fungus has grown around it in that bottom right picture there. So you're going to find this one on dead hardwood trees. Looks like it's present mostly year round. Not sure we're going to have any luck finding it, right now, but it's one that we might see throughout the mushroom season this year. Okay, we're doing pretty good. How's everybody doing? You guys okay? Should I keep going? Yep. Okay. All right. Cool. I'm gonna. I think we're about 2/3 of the way there. So we're gonna move on to the cup fungi, and with the cup fungi, I tucked in the jellies because this diagram didn't necessarily have jelly fungi broken out as their own group. So this is one that is, I think, probably, again, relatively common, but maybe we just have not done a good job of documenting it. So we've got Ascocornea sarcoides, known as the purple jelly disc. I also saw it labeled as purple jelly drops in some of the identification guides. Small, as you might see based on the sort of size of the moss there. It's another one that has an interesting two-form body. So in one form, the cup form, it's actually producing spores that are gonna, in this case, be the products of genetic recombination, so the sexual stage, versus this more sort of amorphous, sort of coral blob-like form that's also gonna produce spores, but those spores are just gonna be basically exact replicas of the genetic material that exists in that fruit body, which is the asexual spore production stage. So small, either purple cups or this jelly body, both having a very gelatinous texture. There's some differences in the spore and basically the characters of the spores that might help in terms of sorting this from other related species. Small enough to kind of why bother on food, be a hard one to make a meal out of. Unknown though as to what it is. I mentioned both of the two different sort of morphs that you have. You're going to find this one growing on hardwood logs. So keep an eye out for the hardwood logs when you're walking around in the forest. If you're bummed out, you can't find some morels, go park yourself, take a rest, sit next to a hardwood log, see if you can find some of these fungi while you recharge your batteries, and you can get back out and get the morels. Warlock's butter is the name for this one, Excidia nigricans. So there's a handful of these black jellies that fall into this genus Excidia. They can be pretty impressive looking. So they're weird sort of blob-like structures. I don't know anything about their edibility. They can be small to actually pretty big. This one tends to fuse together into this sort of larger combination of what are likely starting out as individual little fruit bodies that have sort of fused into this bigger blob. It seems like there's some issues with identifying this correctly. There's some other close relatives. So Excidia glandulosa is one that's in the Mushrooms of the Midwest book, and there's actually a number of records of it in iNaturalist. Excidia nigricans, in this case, it looks like they're different in that this one tends to be sort of bigger, and you get these sort of larger collections rather than sort of individual independent, in this case, fruit bodies. And this one's distinctive. It's got sort of a dotting that exists on the outside of its surface. They've mentioned that it would actually be interesting because you'd think black mushroom, what color would the spores be? Turns out the spore print off of these, if you got one, would be white. So find this one on dead hardwood branches. And again, we should be able to find this one hopefully in the fall or winter. Okay, so moving on to some cup fungi. So these are ones, again, if you're a morel hunter, keep an eye out for beautiful red cups that are in the same habitat that you're looking for morels for, because you might have a good chance of tracking down a couple of these different species. This one is Sarcosypha ostrica. Sarcosypha ostrica, the scarlet elf cup, actually looks very similar to Sarcosypha dudleyi. So I'm going to double up on this one and try and indicate what's different about these two here. So both of these are relatively small-ish cups. They're not massive, smaller size, but bright red, so quite apparent, standing out, in the leaf litter. Pale undersurface, usually don't have a stem, so the cup is basically all you get, and then it's just touching right onto the soil. It sounds like to get these two to be differentiated, we need to go under the microscope. But when you do, it looks like you'll be rewarded with some really beautifully different spores. So you can see the spores in the upper of, in this case, Sarcosypha ostrica. And that one is, it's got little teeny droplets. It has no sheath on the outside. It's also got these funny, curly, very hairy, curly Q cells that are on the undersurface. Versus Sarcosypha dudleyi has this sheath. It has big individual, in this case, 2 droplets, oil droplets inside of the spore. And so it seems like if we got these two and we just looked into the scope, we'd have a good chance at distinguishing these two from each other. Both are going to be typically associated with down, dead branches, logs, sometimes buried. We might also see Sarcosypha dudleyi at a different season based on what the records I could track down versus the other scarlet elf cup. So keep an eye out for these beautiful red cups. They're really pleasing to the eye. They're quite striking when you're out in the woods in the spring. This one's going to be tough to see. It's teeny, but if you're paying attention, like I said, if you're next to these logs and you're looking around, you might find this beautiful eyelash cup fungus. So this is Scutolinia. Some of you may know the relative of this one, Scutolinia scutellata. This one is Scutolinia setosa. I loved what Michael quote. His quote on this was just fantastic. So I'm going to read it in case you can't see this on the screen. It says, "Have you ever tried to take a nap on a log? ? You doze for a few moments, then you suddenly wake up and you find you've turned your head. Your nose now rests squarely in a knothole. The knothole is filled with decay and dirt and a bunch of teeny orange things with ants crawling on them. I probably never would have seen Scutolinia scutellata the first time if nature hadn't shoved it up my nose." So that one is small. This is even smaller. So these are just absolutely teeny little orange cups. But if you get your hand lens out, you get your little, in this case, either a hand lens or your little microscope lens, whatever you need to see these things, you'll see these beautiful erect brownish black hairs that are present on the outside of the cup. Coloration wise, this is sort of not yellowish. It's more on the orangey red side. So that'll separate it from another Scutolinia species. Again, you'll find these growing right on logs, is at least where I've seen a lot of these before. Okay, so moving on to the last group of mushrooms here. So we're into, this is sort of a little bit of a catch-all bin. So the coral fungi, I threw in the clubs and fans. So this one, Spirasus americana. So some of you guys have heard of Spirasus, the cauliflower mushrooms. These are some prized edibles for those who are interested in collecting. Interesting, Spirasus is a genus that's gotten some good sort of study from in this case, scientific perspective about trying to sort out which species are here in North America, which species are in Europe. This is a big mushroom. It can be really quite large. Turns out that in most field guides, the name Spirasus crispa is applied. That's a name that is belongs in Europe. And so they've come up with some different names. In this case, we probably have Spirasus spathulata here in Minnesota as well. That's I've seen in the field guides. It looks like the big difference, if you look at these things, has to do with the fans, the kind of characters of the fans, their thickness, as well as the kind of zonation pattern that you see on the fans of Spirasus spathulata that you don't see on Spirasus americana. These can be big, as you can see in the figure legend. Some of these have been reported to be up to 50 pounds in weight, which seems enormous. But in theory, you can come back and find these on a regular basis. So once you get a Spirasus tree, mark your spot so that you know where it is. Because if you get a good Spirasus tree and you like what it tastes like, you can go back to that spot for hopefully many years to come. So that's one. This was another one. I'm going to be totally honest. I had never even heard of this genus before. I really didn't know this one at all. It's called the cabbage head fungus, according to iNaturalist. So this is Diomyces phillipsii. Here's a picture of it. It looks like it's fruiting out of right next to somebody's, what looks like cement or some sort of garden block. Can't really tell what's going on there, but you can see for size a finger in that picture. This is one where I'm really not sure. The last two images, I'm convinced maybe those are actually a different fungus. This may be here. Finding out about this fungus was interesting. It was described from the Czech Republic back in the early 2000s. If you look at this one and you look at what the Czech Republic collection looks like, there's some differences, right, in terms of kind of what the coloration is, or the amount of kind of gelatinousness maybe is important. Whether it's forming, I don't know if you can see my cursor or not, these tube-like structures, I think is a character that may distinguish this from, I don't see many of those same tube-like structures on the Deomyces phillipsii from the Czech Republic. Similarly, if I look closely at that Sporasus americana, I'm not 100% sure that looks like necessarily the Sporasus americana pictures that I put up before, or that I've looked around on the web to see as well. I think there's a reasonable chance that both of these might actually be a species that we have here that's known as Tremella reticulata, which we also don't have in the Bell collections. So I'm happy to have any of these collected and included in our collections. We don't have a single Sporasus in the Bell collections. I should have mentioned that. So whether you find Spaculata or whether you find Americana, please, by all means, feel free to, you don't have to give up all your collection. We'll take a small piece of it. You can have the rest of it to enjoy and eat if you'd like to. So you can read a little bit more about this species. Again, Michael Kuo's website is just a fantastic resource for us here in the Midwest. He's a collector in Illinois, an expert, and he knows a lot about the mushroom fungi of the Midwest. And so I've used his website as a handy resource to help us with some diagnosis and identification. So I put together one last slide that I hope gives you sort of a sampler menu of maybe some of the key fungi to look for as you're out in the woods this spring, summer, and fall, when you can find some of these. There's some replicates because there are maybe different seasons that we would see these mushrooms, but it would be awesome and much appreciated if any of you who are here are willing to keep an eye out for these fungi. And when you find them, get in touch with either folks through MMS. So that could be Ron Spinoza, the current ID expert for the club. It could be, Tim, it could be John, it could be Kathy, Nan, whoever it is who's affiliated with the club. They can help with, again, getting in touch with me. You're welcome to email me directly and tell me you've got a collection that you're interested in and excited about. I'm happy to try and work with you to see if we can get an identification for it. And I think that's it. That's all I've got.
Does anybody have any questions for Peter? Peter, maybe a couple of questions, if we have any. I think everyone's just really excited to get out.
I was on my own virtual foray like all weekend when I was putting the slideshows together. So that was great. It was self-serving. So yeah, I'm with all of you. I want to be out in the woods.
Can I just say, I think the organization you did on those slides is terrific. I mean, what distinguishes what doesn't, where there, I mean, it's like a perfect amount of information to kind of carry with you when you're out there. So thank you.
Yeah, I wonder if.
So Peter, excuse me.
Oh.
If we make a collection, should it be photographed on iNaturalist then, or?
You are welcome to put it on there, Ron. That would be great. I mean, I think it's might as well.
Okay.
It is a place where I think there's more and more collections and observations. That said, if you make a photograph, we'll take that and include that as some of the information we can add to the Bell Museum collection. So we don't have good field photographs for as many of our collections as I wish we did. And so I would love to have that be something that we could, if you made the collection, you had the field notes and you had an image, if you could share that with me as well, we'll make sure that gets onto the Bell Atlas. That would be great.
Okay.
Tell us about the specimens you'd like to have in hand.
Any of those would be fantastic. Is that the in terms of in hand? What do you mean?
Physical specimens. How do you want them delivered, dried, whatever?
Yeah. Gotcha. Thanks. So that's a great question. I think that the best thing you could do is, so if you see these and you think you've got one of these on the list, by all means, take some field pictures, make a good collection. Again, take some good notes. What's your habitat? Ideally in your phone, there's an image that has a GPS coordinate on it. But if you happen to have that latitude and longitude, that's very helpful. You're welcome to dry those specimens down at a very ideally low heat. So don't stick them in your oven. If you can let them basically air dry in a nice sort of even heat, they'll desiccate just fine. We will have to, at that point, to get them into the bell collections, freeze them for a certain period of time to make sure they don't have any insects on them. But that might help in case we want to do some molecular work on these later. So you can dry them down. Once they're dry, then you can go ahead and stick them in pretty much any container, but I'd recommend, honestly, something that's still breathable. So paper bags are great as a storage location. If you put them in plastic bags and they're not all the way dried down, they can start to get moldy, and that would be a bummer to have, basically almost a collection and then have it all covered up with a different fungus. So, yep, you can also deliver them fresh. We can do all the drying in my lab, and we can get them into the collection that way. But the main thing is just as many notes as you can record for us with that information from where they are in the field. That would be great, and then we'll take it from there.
Fantastic.
Sometimes the daunting task is how to actually physically get them to you. So suggestions.
Yes. Good question. So I'm hoping that there's a reasonable chance maybe we'll get back to some in-person MMS meetings. And if they are, I live very close to where the MMS meetings were held on the St. Paul campus. And I should be, available and on campus. This is Twin Cities St. Paul campus pretty regularly throughout most of the year. And so that's a possibility. That said, I've driven out and gotten collections from folks that are in the Twin Cities. I tend to move around a fair amount looking for fungi in various places. And so email me, and you can email me and we can figure out how to, if there's a common drop-off point that works as well. Or again, these can be held for a period of time. So you could bring them in, you could bring them to the club, and they could be held by Ron or John or Nan, whoever, and we can get them that way as well.